Welcome to a mini-series of Neurosalience, featuring interviews with some of this year's annual meeting's keynote speakers. Dr. Max Shine is a systems neurobiologist at the University of Sydney, Australia. He uses functional brain imaging to understand the mechanisms of cognition and attention, both in health and disease. He has particular interest in understanding how the different arms of the ascending arousal system modulate the organization of the brain to facilitate adaptive behavior. We're delighted to have Dr. Shine give one of this year's keynote lectures at the OHBM annual meeting in Seoul. So in this interview, I'll chat with Mac about his research, how he got into science in the first place, his attitudes to academia and mentorship, as well as where he sees the future of human brain mapping. I hope you enjoy.
Thanks very much for doing the interview, Mac. It's great to have you on.
Thanks very much for having me.
Yeah, let's start off with just a very broad, brief overview. If you could describe your research to someone who has little expertise in this field, how would you describe that?
Yeah, I actually spend an awful lot of time trying to figure out how to describe what we work on. I consider myself a systems neuroscientist. I'm really interested in how all the different parts of the brain work together to give rise to particularly cognitive function, but we don't always limit ourselves exactly to that. And to do that, we use a set of different tools depending on the type of question we're asking. Oftentimes, functional imaging is a huge part of that, and fMRI in particular. But we've recently been really interested in the kind of benefits we can get from using computational modeling, so building biophysical principles into a little toy model and seeing how well we can recreate some of the features of neurobiology, but then get them to match the kinds of neuroimaging signatures we see in things like fMRI. And that is, as you could imagine, a sort of endless, ginormous task, because the kinds of models you can build, there's so many different parameters, so many different choices you have to make. So I view that more as the kind of process that we use to develop our understanding and to test hypotheses about what we're seeing. So the reason I really love that, though, is that I grew up in the field of functional neuroimaging and really enjoyed a lot of the answers that I was getting from functional neuroimaging, the kinds of network level interactions that you could use to explain something like working memory. But I also personally absolutely love neurobiology and love reading papers about the organization of the nervous system down at the cellular level. And I feel like there's just such a massive disconnect between those different scales. You've got this macro scale vision of the brain as a whole, and then you've got this micro scale level detail. And biophysical modeling to me is a way that you can start to make some form of contact between those. It's not without its foibles, and it's definitely a slow path, but it's a really rewarding one when you can create some form of principled model that's based on what you're seeing in the neurobiology, or what in fact other scientists are seeing, and then use that to try to understand better the kinds of neuroimaging signatures that you're seeing from your experiments.
I wonder if you might be able to give some sense of how it is you've got here. Basically, I got the sense that you're not, you're doing a lot of this modeling, but you haven't got a mathematical background necessarily or anything like that. So how is it that you got interested in this topic, and what was it that interested you in a scientific career at all in the first place?
Yeah, lots of different ways to tackle this question. I think I've always idolized science. My father's an academic and my uncle's an academic, and so I kind of grew up with that as a kind of demonstration to me of how satisfying of a career you can have if you really pour yourself into a deeply fascinating problem that's just, orders of magnitude bigger than you are. You can essentially forever be curious, and I think I was always really deeply attracted to that. I didn't particularly like working on a bench, though. So in undergraduate, I loved biochemistry, but I really didn't like pipetting things. And so I ended up, after a year abroad, I went to medical school, fell in love with the idea of understanding systems there, really trying to understand how you could have a physiological level description of an organ system like the heart, or think about the heart in the context of the whole body with the cardiovascular system, interacting with the kidneys as well as the lungs, as well as the brain, and thinking about how that system could work or fail was really deeply satisfying to me. But as soon as you tracked up above the neck into the brain, all of those stories just sort of dissolved like dust sort of into the wind. And there were lots of more simple stories, so what stories that you could tell, but they weren't grounded in the same way they were in the other systems. And so when I left medicine and did a PhD, I was originally working in Parkinson's disease, doing a lot of systems level work in imaging, trying to understand how some of the more complex symptoms of Parkinson's arose, the ones that didn't really make as much sense in the traditional models, by thinking about this sort of systems level perspective. I found that really, really satisfying, because now you could come along and say, what kind of a physiological description could I give of this system? And then how might it look at the imaging level? And more than anything, I kind of learned, I enjoyed those types of questions, but I was really dissatisfied with the quality of the answers that I was getting. So I did a postdoctoral fellowship, worked with Russ Poldrack at Stanford, and he was just transformative for my career, in the sense that Russ really showed me, through the process of learning how to develop my scientific questions and develop the kinds of evidence that I was willing to consider as helping to adjudicate between different hypotheses, really showing me how to conduct myself as a scientist in that space, was so helpful for me. So that when I came back to be able to run my own group, I felt that I wanted to sort of really double down on those kinds of principles that Russ had instilled in me, but ask mechanistic questions. And so hired some people in my lab that had a background in modeling. One physicist, Eli, had a background in mean field modeling, and then another, Brandon, had a background in kind of microscale modeling. And together we kind of decided to tackle this really big problem of how do we build principled models that sort of make contact with the microscale and the macroscale, but also match neuroimaging. And the really great thing for me is that in that triumvirate, even though I'm quote unquote the person hiring Brandon and Eli, they're actually kind of like my boss when it comes to me understanding modeling and understanding physics and understanding the microscale, and spiking neurons look nothing like the bold signal. So I got to transform back into being a student again. And so it was almost a little bit like I did another postdoc in modeling. But together, I think, we've really come up with some really cool ways to think about the system, new kinds of techniques that I never would have thought of on my own. And it's really a part of this really deeply collaborative group that is forming in Australia. And I really love that whole process. So not a particularly linear path, but I think at the core of the whole thing is this really deep curiosity for coming up with simple explanations for otherwise seemingly kind of irreducibly complex functions.
In 2016, you published a paper in Neuron, describing how the brain seems to switch from these states of segregation to integration, and describe how that relates to behavior. Just for people not familiar with that work, can you unpack that a bit and try and give a sense of the impact of that paper with respect to the context in which it came out, and what were the main findings of that?
Yeah. So that was a really fun project to work on, and I think represents a really key moment in my scientific upbringing, where I kind of came from a world of kind of clinical neuroscience in my PhD towards this sort of much bigger ocean of cognitive neuroscience and neuroimaging and network science. And so a lot of that was me learning how to conduct myself on that scale, and to do work that was defensible from just many, many different vantage points, which I think was a really great thing for me to go through as a young scientist. The particular project itself was, it came around at the time when people were starting to play with this idea that time averaged connectivity matrices were informative, but maybe missing some interesting fluctuations on a shorter time scale. So the field as a whole had transitioned a little bit from doing task-based analyses and fMRI towards the resting state, I think, which was very helpful for us to think about this system that was at the core of everything, force you to think outside of a thresholded statistical map towards this, living, breathing thing in front of you. But the understanding of how that related to tasks was still kind of being developed. There were some really beautiful papers. Michael Cole had a really lovely paper, and this really cool idea of task-based reconfigurations, which I was really inspired by. And I'd been reading a lot of work from Olaf Sporns about network science and how to think about, if we pretend that the brain is a graph, set up a bunch of little nodes, little connections between them called edges, and then just try to describe that in a data-driven fashion. What can we say about it? And it turns out you can make a lot of really interesting inferences about the network as a whole using graph theory. And so I was really interested in trying to combine these things together. So this dynamic perspective, the idea of task-based reconfiguration, and then this network neuroscience approach. And so Chris had the idea of going back to this old paper from 2005 by Gumer and Amaral that characterized a network in a particular way, where you kind of take a network of all these little edges, do some clustering on it, and then afterwards ask each of the nodes how well connected are they within their little local community in their cluster, and how many of them are sort of sitting between the clusters. You can imagine something like someone who hangs out in their own department at university versus someone that wanders between physics and neuroscience. And so what we found was when we just tracked that measure over time, and I don't want to pretend like this was a linear path, because we beat our heads against the wall for a long time and clustered things in crazy ways that just didn't fit the data. A lot of this was just discovering the right way to look at the data. But if you just step back from it and do a joint histogram of these different values and plot them over time, you see there's just beautiful fluctuations back and forth between these different extremes of really, really interconnected networks and really sparsely connected networks. We call them integrated and segregated networks. And it turns out that if you then go and do a task, you shift into this integrated network. And then if you go back into the rest block, you go into these segregated states. And so we were really excited about, this whole perspective, just because it gave us, a different way to look at the system, rather than thinking about, is the default mode network on or off, or is the frontoparietal network loading in or out, or connecting to the dorsal attention network to let you do something. You could now say something a bit more fluid. Is the system ready to do some interconnection, or is it now in a state where it's more protected from blending? And I just found this to be a really beautiful idea. It's one of these ones where once you see it once, you can't help but see it everywhere else in the world. You start to think of, your kids' school group as segregated or integrated when they're playing on the playground or going to their classrooms. And it's this sort of new set of lenses to which to look at the whole world, really. It took us a really, really long time to get this published. And I actually got what I'm going to call the best rejection of my career. So when this project was about 2/3 of the way through, we submitted the paper to eLife, and this is just around the time that eLife had kind of come on board, and we got a couple quite nice reviews, very constructive, positive reviews on the paper. But the editor at the time, Tim Behrens, actually wrote a personal email and rejected the paper. And his reason for rejecting the paper was that he said it's not quite done yet. You've got something there. There's some system going on. There's some cool measurement you've got, but I don't understand why is it doing it. Your job as a scientist is not just to show a thing, it's to test hypotheses about what it could be. And I remember at the time being a young postdoc being really kind of grumpy, like, oh, how dare he reject my paper? But then the more we thought about it, the more we talked about it internally, like, wait, this is actually a really fascinating question. What could this be? And so just flipping the question from how quickly can I publish a paper to what is going on here opened up a huge world for me. I ended up writing a review paper with Russ that went into a special issue of Neuroimage, about network level reconfigurations and like what could cause them. That was just so much fun because I got to read about neurobiology and cognitive neuroscience and different types of pharmacological agents and just think about the possibilities, which is so much fun. But I also got to then go around the Stanford campus and meet with all these different network specialists. Like I met a guy that specializes on social networks, Yura Leskovich, and asked him what kinds of social contexts can change this integrated segregated feature. And I met an economist, Matthew Jackson, and I was like, oh, Matthew, if you like changed the the rebates or something, or if you like messed around with the inflation, gave people more tax returns or something, would that change whether or not they spent money and then did this thing? And he's like, no, not at all. That's silly. But he's like, surely in the brain there's regions that project everywhere and just change how well the different regions can connect and communicate with one another. And honestly, it was like someone slapped me in the face with a wet fish, man. It was like, oh my God, it's the arousal system. That's what's doing this, right? And I wouldn't have come to that hypothesis without being rejected, told to go look, gone and looked and been open and curious and being told I was, silly by a bunch of professors at a leading institution in the States,
I guess
right?
it's a message of it's better it's better to go to do deep papers rather than just do as many like try and get this shallow one out as quickly as possible. Because as soon as you dig below that layer, you seem to have
Yeah,
opened up this like
it
treasure
opens a
trove of.
chasm of questions, right? And now, in many ways, the rest of my career has been defined by that junction point of giving into the curiosity of the hypothesis driven science. Like, if it is this, what would it look like? And that one simple switch now means that I get to collaborate not only with brilliant neuroimagers around the world, but with cellular physiologists and people that are stimulating the locus coeruleus in a anesthetized rat while measuring the bold response and seeing whether or not the kinds of causal manipulations of this circuit cause the kind of signatures that we see when we look in a human. So that's work with Valerio Zerbi ongoing at the moment. But that's just one example, right? And I feel like in many ways, just taking that step into uncertainty has been so rewarding for me as a young scientist, because it means that the kinds of questions you can answer afterwards are so much more interesting and so much more like you can relate them back to this curious question you had. And I wouldn't have got that without being rejected. So that's why I say it's the best rejection I ever got.
Not every rejection is a bad thing, right? It's
Yeah,
like,
exactly.
but this, I
100%.
mean, this is a whole other branch of what you do, which is obviously related, and you've just touched on it. But um, maybe you can, if you could offer a little bit of description on how then this leads into your work on neuromodulatory systems. How are these states influenced by those, and what aspects of these neuromodulatory systems interest you?
Yeah, so we had this I had this kind of epiphany after talking to Matthew Jackson about the arousal system, but then that's when the real work begins, right? Because then you need to start asking, how can we even test this? What kinds of levels of evidence would we accept versus reject? What is our understanding of the arousal system and what that means and what it is and isn't? And I feel like, there are so many different layers to that that cake that I'm still just, eating the crumbs on the edge. It's such a huge problem. So the two kind of arms that we went down, one was doing pharmacological fMRI using open data sets. That gives you some really interesting answers that are oftentimes a little more non-linear than you'd be comfortable with, just because every time you turn up a neuromodulator, there's all these compensatory effects that turn down the firing of a particular nucleus, and it gets really messy. And we also did a bunch of computational modeling. And the long and short of it is that we're still really learning about the best ways to do this kind of analyses, the best kinds of measures to use. The one that we're really attracted to at the moment is related to the network idea, but it's got a slightly more dynamical bent. What we do is we conceptualize the whole brain state. Let's say all of the bold signal across all the voxels or the parcels that you'd have at a given time point as a brain state. Like many people in the field are doing, and then we ask about how that brain state evolves over time. So we imagine it as a ball rolling around a kind of hilly landscape. And what we think of as the neuromodulators as changing the kind of topography of the landscape. So making it flatter, making it easier for the ball to roll to new locations, or making it more rugged so the ball could roll into a different state and stay there and stay sort of stuck. And so we think of things like noradrenaline and acetylcholine, these sort of cooperative but opposing neurochemicals, as kind of changing the balance between the flatness and the steepness of this state of this landscape. And we're going about trying to test these hypotheses with causal imaging, with modeling, with different kinds of techniques to measure these kinds of features from neuroimaging data. And that's an evolving process. There's some really exciting stuff coming out soon in peer review at the moment, looking at some really cool existing data sets. David Leopold's group with Katie Chang, Juanita Turchi had this beautiful paper in 2018 in Neuron where they inhibited the nucleus basalis of mainnet, the main cholinergic input to the cerebral cortex. And we were able to get that data from Katie and David and Juanita and have run that analysis, our analysis on that data, and we can essentially recapitulate their original result, but also accommodate our way of thinking about the effect of acetylcholine as this kind of stabilizing effect. And so to me, this is like another, again, deep example of the kind of benefits of collaborative open science, right? We couldn't have answered these questions on our own. I don't know the first thing about recording data from a macaque or the kinds of challenges that go with that, but here's this great group that deliver us this beautiful data set, and together we can get a more satisfying answer that now interconnects their beautiful results with this other perspective that now lends itself to modeling, lends itself to other types of questions and other types of curiosities that come out of it.
Typically, when we in a lot of fMRI research in particular, it's very cortex centric. I guess, why do you think that is, first of all? But what are the benefits that we're getting from looking beneath that surface? You're cutting out a whole lot of the brain by just looking at those. You're cutting off brainstem, you're cutting off cerebellum, all these subcortical systems. Do you think there are other viewpoints or practices that are sort of right there in front of us that we're doing that maybe are hindering our progress in neuroscience?
Yeah, it's a great question, and there's lots of different ways that you could tackle an answer to it. The historical one is fascinating for a really fun reason. Well, it's maybe a bit macabre. The primary physician for the Roman Empire around the time that the Colosseum was really just churning, Galen, was one of the first to notice that different gladiators that came in with a different part of their cortex smashed in by an axe or something had different deficits. So he noticed if their occipital lobe got smashed on the right, they couldn't see anything on the left, and if their frontal lobe on the left got smashed, they couldn't move their arm on the right or something. And he was one of the first to kind of really point towards this idea of cortex as functional localization. And there's obviously been many others over history, but I think that's an interesting starting point. I think the cortex is a fascinating object. It's really expanded over evolutionary time. It's also expanded in its complexity. If you look at the pyramidal neurons in the frontal cortex of a human compared to a macaque, compared to a mouse, there's actually more elaborate dendritic trees and different kinds of weird synapses and boutons and things like that on these cells. So the complexity of the cortex is alone a fascinating problem to solve. I think people have been really attracted to this idea of the cortex is right there. I can see it. It's really big in humans. If I remove part of it, there's an obvious functional consequence. And it's also, because it's on the outside, a little easier to measure, right? The signal to noise, if you just go through the surface, a little easier than the deeper structures. And so I think you can ask a lot of great questions there. And I think that part of the reason, it's a little bit like, the low-hanging fruit, but here the low-hanging fruit's delicious, right? The berries are tasty and they're bountiful, and there's many more of them than we can eat. But if that's your goal, then I think it's absolutely a great way to go about what you're doing. I don't think there's anything disparaging about being a corticocentrist. The cortex is beautiful. I work very closely with corticocentrists and love discussing things with them. But if your goal is to understand how the system functions, or how to make contact between different species, like how it is that your mouse model of Alzheimer's disease might be linked to your human model of Alzheimer's disease in the patient right in front of you in the clinic, I think the challenge for you is to ask, how could I make a correspondence between these two? And it turns out that subcortical structures are just, far more conserved, and for lots of good reasons, in that if you remove them, it's not just your inability to see the left part of your world. If I knock out your brainstem, you're either in a coma or you're going to die. And so these base level functions that just let us be a kind of aware animal embedded in our world are much more conserved and deeper in the structure of our brainstem and thalamus. So I think understanding how the system works as a whole, and also the functional consequences of these things, is deeply important for certain types of questions. And I personally just love thinking about these kinds of things. I love looking at something as weird and wonderful as the cerebellum and thinking, what on earth is this thing doing, right? It's half the neurons in the brain. It's receiving all these efference copies from the cerebral cortex. It's feeding back to the thalamus. What's going on? And just asking these questions and trying to think about what the impact of that circuit would be on this beautiful cortical sheet that's sitting there. If this cortical sheet's like doing some algorithm, let's say, let's pick your favorite algorithm, some kind of predictive coding or something. Surely, if it sends a message, quote unquote, down to the cerebellum, and then it comes back by the thalamus and hits the cortex again, that's going to have some impact on the kinds of messages it can pass, or the kinds of errors it would receive, or the kinds of ways that it could update its model based on the errors that it received. So to me, you're just going to get more nuanced and really enriching questions out of taking a systems level perspective and thinking about the implications of that than you will by just looking at the cortex on its own. So I don't know if that's a nuanced enough answer for you, but I think corticocentrism is great, but it's really helpful to think about the rest of the brain if you have different types of questions.
Yeah, it's very well hedged. No, you're not making anyone angry with that answer as well. That's good.
But it's because I believe that if we're going to solve whatever it is that the brain's doing, it's going to be a community that does it, right? It's going to be a whole bunch of us with different levels of expertise finding the language to work together. That's
Yeah.
going to solve it. It's not going to be, some person sitting in a room somewhere that goes, "Ah, eureka, I solved it," right? That's
Yeah, or
silly
my
to
way is right and your way is wrong. And
Yeah, let's be done with that. And I think the HBM is a great community for this, right? Because we have baked into our core this really strong set of open science principles and very collegiate atmosphere, that I think it's actually a great place to conduct one science as a young student interested in these different concepts, right? If you really want to understand how to do multivariate statistics at a high level, there's tons of people you can talk to. If you really care about neuroanatomy, there's a ton of people you can talk to. If you really care about integrating MEG and EEG and fMRI, there's a bunch of people you can talk to. So depending on where your question goes, you've got this sort of inbuilt network of brilliant, really open people to talk to. And I think the collegiate atmosphere to me is just like emblematic of why I would kind of back this field to make really great progress in the next, 10 to 20 years.
well, on that specific note, actually, yeah, I wonder if maybe taking a step back to bigger picture, where do you see this field going in the next, I don't know, 5, 10, maybe 20 years? Where do you see the field and then your work within that?
Yeah, that's a really great question. I like to tell my kids that anytime someone tells you they can tell the future, they're either lying to you or trying to sell you something. Um, so my first comment is going to be that I don't know where I see things going, and the thing that gives me a lot of excitement for the future is seeing a lot of the disciplinary boundaries breaking down and seeing conversations between people that have expertise in neuroanatomy and statistics and modeling coming together and having conversations. I just recently was on a call with Nick Palomero Gallagher, whose work I absolutely love, and Sean Frutis Walsh, who gave a couple talks during the pandemic for us and just blew our mind. He's worked with Xiaojing Wang, does really beautiful anatomically inspired neural modeling. And we got to be in a call together with Carmen Salvato, who does beautiful thalamic work. And it was just fun. At the end of the call, we were like, how great is this? We get to have this conversation together over Zoom. We're on different parts of the planet having this conversation about this beautiful intersection between these completely different fields with different constraints but different benefits. And to me, that's the really exciting thing that is starting. You can start to see it happen in real time. In terms of my own work and what I want to do, I want to keep being curious and keep following my students' questions. We have a real, I think it's both spoken a little bit, but mostly unspoken policy in the lab, which is we want to find out what you're interested in. So when students come to the group, we try to find out what it is that's the burning question for them. And so while that maybe kind of makes the nature of the group a little chaotic in terms of the questions, I think it leads to really deeply satisfying explorations. I don't know what we'll end up working on in 10 to 20 years because I don't know what students will join and what kinds of things they'll be curious about. But I think there's so many great questions out there for young neuroscientists that if you can take this interdisciplinary approach and develop these satisfying questions to ask, I think it's going to really open up some cool inroads.
People will be interested to know um, about you as a PI and as someone who has um, gone through PhD postdoc training and then had to set up a lab. How was that for you, and how would you describe your mentorship style?
Yeah, it's a really great question, and I think it's one that's ever evolving. I played a lot of sport when I was younger and was fortunate to be part of a lot of really successful sporting teams. And the thing that I think characterized all of them on the years when we could be successful was that there was this lack of individual ego and a kind of building towards something that mattered more for the team. And so uh, I my mentoring style is essentially trying to recreate what I saw as those really successful team-based dynamics in my experience in science. And so we work really hard to make sure everyone's voice is heard. Work really hard to make sure that people are working on problems that are well suited to what they can do and what they can bring to the team. We work really hard to make sure that we're finding people's excitement and curiosity and energy for science so that we don't have to worry about that part of it. That was a mentor of mine and collaborator, Ethan Scott, gave me that advice during the pandemic. He said, don't worry about whether the project's almost done. Just worry about whether your students are excited to go to work. And that was really transformative for me as a mentor, because I can help with the excitement part. I can show you what I think is a cool thing in what you're doing. I can help you realize that responding to the really grumpy reviewer too is actually going to make your science better, and it's going to help you communicate your work better. Talking to my postdocs at the moment who are in that stage of writing grants the whole time, guess what? Writing grants sucks. It's really annoying. It's frustrating. It's inevitably going to fail because only 10% of them get funded. But if you can, through that process, find a way to find the right analogy or the right way of framing your problem so that it's so clear to someone and they get why you work on what you work on, it's worth its weight in gold. And so I can find ways to motivate my team to enjoy the parts of this that are not as naturally enjoyable. And to me, that changes everything. I can become a player on the team, which is what I really like to do. We believe in a really flat hierarchy in my group where different people would run lab meeting. Whoever's the person leading the project will take the mic, in other words, will lead the project. Oftentimes my postdocs will run off with students and do things together that I couldn't understand, because there's no point me slowing down that train while they're working on modeling stuff. But then when they come back to me, I make sure they can explain it in a way that I can understand so that it can be integrated into the whole. And so we do, and the only reason that this works is that I have a brilliant team of just really selfless, curious scientists with me that have kind of like bought into this notion of a team-based approach. And it's not without its lumps, science has this stupid way of attributing linear credit, first and last author get all the credit on a paper or something like that, whereas a lot of times people that are actually doing the science know it's the middle authors that have been the critical part of the whole thing. And so it's really difficult to kind of like navigate that space, and we don't know how to do that properly yet, but I think just having open conversations is the best way.
Amazing. Do you have any advice for early career researchers who are trying to establish themselves within this field? It sounds like, you've learned like a lot as you've gone along, and that's great. But people coming in, is there like one aspect of the field that you think is most beneficial? Like would they study physics or biology or computational modeling, or do you think it is just a case of being a jack of all trades, or having one thing and then learning as you go? How would you approach that?
That's a really, really great question. When I was a junior PhD student, I remember feeling, and I still do in a lot of ways, like we mentioned before, I think you have to get really comfortable with being wrong and almost embrace it, like run towards it. Because if you can find the places where you're wrong and learn about them, you're now no longer wrong in that area in the way that you were wrong before. You could be wrong for a variety of different reasons that you haven't figured out yet. But having that growth mindset, I think, is super important. And I think that probably is more important than what it is you study in particular. I don't think there's going to be one magical, vocation or training that will kind of get you, closer to the end point of this game than any other. I think coding skills and coding proficiency is an absolute deal breaker. If you can't code at a level that you can both believe your own results, but also convince your colleagues that what you're doing is reproducible, then I think that's almost like a ticket to the club nowadays, I think. But then once you have the kind of basic coding proficiency, understanding of stats, the world's your oyster in a way. And what I would encourage students to do is figure out what the thing is that you like the most. Do you really find an answer based on physics principles that are going to be abstract and lack detail really satisfying? Or for you, do you really care whether or not this pyramidal neuron and not that pyramidal neuron is the one that's important for the behavior? Or do you really, really care about whether or not this brain region or that brain region, or this cognitive task or that cognitive task, or whatever? Find the part that really speaks to you and seek out the people that can help you do better at that. Be less wrong in that space. and you'll naturally find the things that you like to do show up to you. So if you have a growth mindset and you back yourself, I think good things happen. But yeah, make sure you can code.
So I just want to wrap this up then. We've got so much good stuff here, but just want to finish off really with whether you're able to give us a little taster of what is going to be the focus of your talk at OHBM. Maybe it's too soon to get details on it or anything, but is there anything that you can tell us about that?
Yeah, I'm still working on some of it, but I think what I want to try to do is kind of, put myself back in the shoes that I was in when I first walked into the poster hall at Quebec City as a young, wet behind the ears grad student, and was just overwhelmed with this beautiful field of crazy network neuroscience and neuroimaging, and think, what kind of answers would I have gotten then to a question I asked, and then what have I learned along the way that I think might be helpful for getting different kinds of answers and different kinds of questions that come from that, and try to take a kind of, a little bit retrospective, a little bit prospective, and speak about some of the dynamical systems, neuroimaging, computational modeling, and neurobiology work that we've been doing in that space.
Excellent. Well, I'm looking forward to it, and I think it's going to be a great talk. I'm looking forward to OHBM. I'll see you there.
Thanks, mate.
All right, thank you.
Welcome to a mini-series of Neurosalience, featuring interviews with some of this year's annual meeting's keynote speakers.